Assalamualaikum warahmatullahi wabarakatuh. Hello, good morning everyone. Welcome to Top Scientist Webinar World Class University with the topic Update Technology in Dentistry. Ladies and gentlemen, we would like to inform you that we will be taking attendance for this webinar. To confirm your attendance, please kindly fill in the Google form in the chat box. We will still waiting for our professor and the moderators. Please stay with us. Thank you. Dear all participants, we would like to remind you that we'll be taking your attendance for this webinar. To confirm your attendance, please kindly fill in the Google form in the chat box. Thank you. All the participants, the virtual background you can get in the chat box. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Hello, good morning everyone. Welcome to Top Scientist Webinar World Class University with the topic Update Technology in Dentistry. My name is Intan. It is such an honor and precious chance for me to be your master of ceremony in this event. Ladies and gentlemen, we would like to welcome the Honorable, the Chairperson of World Class University Team, University of Sumatra Utara, Dean of Faculty of Dentistry, University of Sumatra Utara, Vice Deans of Faculty of Dentistry, University of Sumatra Utara, Director of RSGM University of Sumatera Utara, the head of Department and lecturers of Faculty of Dentistry of University of Sumatera Utara, the head of PDGI Cabang Kota Medan, all of the alumni, Faculty of Dentistry of University of Sumatera Utara, and all finally the doctors and participants. Ladies and gentlemen, we are honored to have three world-known speakers here with us today to share their insights and experiences regarding our topic, Update Technology in Dentistry. The first session, we have Professor Dr. Tanapum Osatanon, DDS PhD from Chulalongkorn University, Bangkok, Thailand, and will be moderated by Professor Dr. Ameta Primasari, Dr. Gigi MDSC MKS SP PMM. The second session, we have Dr. Dan Johan Apps, DDS PhD Paid Dan and Special Needs MSc DMFR from Artevelde University of Applied Science Gent, Belgium, and will be moderated by Dr. Gigi Cekdaramanja, SPRKG Consultant. The third session, we have Professor Dr. Nicola M. Grande, DDS PhD from the Catholic University of Sacred Heart, Rome, Italy, and will be moderated by Dr. Gigi Dennis, MDSC SPKG Consultant. Ladies and gentlemen, now we would like to start this webinar with the national anthem of Indonesia Raya. Please have a seat in a good position and perfectly. Thank you. Now, this is our rundown for this webinar today. And next, we are going to hear from the Dean of Faculty of Dentistry, University of Sumatera Utara, for her welcoming speech to Dr. Gigi Esi Oktiara SPKGA. Time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, and I hope everyone always in good health. The Honorable Professor Dr. Engineering Himsar Ambarita, as head of the World Class University Program. The speakers, Professor Dr. Tanapum Osatanon, PhD, Dr. Den Johan Epps, PhD, Professor Dr. Nicola M. Grande, PhD, Vice Deans, First, Second, and the Third Faculty of Dentistry, Universitas Sumatera Utara, the Professors of Faculty of Dentistry, Universitas Sumatera Utara, and all webinar participants. Alhamdulillah, for the grace of Allah, we are able to gather in today's webinar. Salawat to our Prophet Muhammad Sallallahu Alaihi Wasallam. Allahumma sholli ala Sayyidina Muhammad wa ala ali Sayyidina Muhammad. I would like to welcome all of you to our Top Scientist webinar with the topic Update Technology in Dentistry. This activity was held with the support of the Universitas Sumatera Utara in the World Class University program. This time, our speaker will discuss about knowledge update in dental stem cells, dental radiography, and minimal invasive endodontics. I would like to express my gratitude to all of speakers. Professor Tanapum from Chulalongkorn University, Bangkok, Thailand. Dr. Den Johan Epps from Artevelde University of Applied Science, Ghent, Belgium. And Professor Nicola M. Grande from the Catholic University of Sacred Heart, Rome, Italy, who have been willing to spare the time for sharing knowledge with all of us. Inshallah, it will be useful to all of us. For all the webinar participants, thank you for participating in today's webinar. And to the committee, I thank you for your hard work in carrying out the webinar. Inshallah, in the future, in the webinar we will have periodically. Akhirul kalam, wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Esi, for the speech. Now, we are going to hear from the chairperson of the World Class University Team, University of Sumatera Utara, to deliver his welcoming speech and to appreciate the Top Scientist Webinar World Class University today. Ladies and gentlemen, we are still waiting to Professor Dr. Himsar Ambarita. Ladies and gentlemen, now we are going to hear from the chairperson from the World Class University to deliver his welcoming speech and to officiate the Top Scientist Webinar World Class University with the topic Update Technology in Dentistry to Professor Dr. Himsar Ambarita STMT. The time is yours, Professor.
Okay, thank you very much for the committee for introducing me. I will deliver my speech on bahasa and Arab in English. Assalamualaikum warahmatullahi wabarakatuh. Salam, Om Swastiastu, Namo Buddhaya, salam kebajikan, dan selamat pagi bagi kita semuanya. Salam sehat, salam sejahtera untuk kita semua. Yang saya hormati Ibu Dekan Fakultas Kedokteran Gigi, Dr. Esi Oktera, Dr. G, DRG, spesialis KGA, para wakil dekan yang hadir pada webinar ini, dan para pembicara serta para moderator kita. Mungkin saat ini tidak hadir bersamaan, tetapi ada seriesnya. Profesor Dr. Tanapum Osatanon dari Chulalongkorn University. Welcome, sir. Dr. Den Johan Epps from Artveld University of Applied Science, Gent, Belgium. Profesor Dr. Nicola M. Grande, PhD from the Catholic University of Stuttgart, Rome, Italy. Para moderator kita ada kakanda kami, Profesor Dr. Ameta Primasari, Dr. Gigi, kemudian Cek Daramanja, kemudian Denis, Dr. G. Kemudian Bapak Ibu, saudara-saudari para peserta Top Scientist Webinar World Class University dan juga tim WCU. Puji syukur kita panjatkan kehadirat Tuhan Yang Maha Kuasa karena atas rahmat dan karunia-Nya kita dapat hadir dalam acara rangkaian Top Scientist Webinar ini. Perlu kami beritahukan bahwa saat ini Universitas Sumatera Utara berada pada Renstra tahap kedua, di mana yang merupakan visinya adalah internasionalisasi to become a world class university. Universitas Sumatera Utara terdiri dari 15 fakultas dan 15 fakultas dan 1 graduate program, dan salah satunya adalah kedokteran gigi. Di sini adalah merupakan suatu kebanggaan bahwa webinar Top Scientist ini, kedokteran gigi telah menghadirkan beberapa pembicara top ya dari tingkat dunia yang ingin berbagi pengalaman dan juga riset terbaru tentang bidang kedokteran gigi. Jadi diharapkan ini akan memberikan impact yang cukup baik kepada academic reputation Universitas Sumatera Utara, dan ini juga diharapkan menjadi suatu pembuka dari kegiatan yang lebih panjang lagi, yaitu bisa riset collaboration dan juga membangun jejaring. Jadi akhir kata kami dari tim WCU mengatakan selamat melaksanakan webinar. Terima kasih kepada seluruh jajaran dekan dan dekanat dan juga panitia yang telah bersusah payah menyiapkan acara ini. Semoga kita semua mendapat manfaat yang cukup baik demi cita-cita bersama menuju World Class University. Thank you, and we wish a good webinar for all of the participants. Thank you very much.
Thank you very much, Professor Himsar, for the speech. Now, I would like to invite Ustadz Fajar Hasan LCMA to lead the prayer.
Assalamualaikum warahmatullahi wabarakatuh. Hadirin yang kami muliakan, mari sejenak kita menundukkan kepala Memohon kepada Allah Subhanahu wa taala agar acara ini lancar dengan baik. A'udzubillahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin hamdan yuwafi ni'amahu wa yukafi'u mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal karimi wa azimi sultanik. Allahumma ya Allah, ya Rahman ya Rahim. Dengan segala kebesaran dan keagungan-Mu ya Allah, tak henti-hentinya kami mengucapkan puji dan syukur atas segala nikmat rahmat-Mu yang senantiasa Engkau berikan kepada kami, sehingga pada hari ini, 29 November 2021, kami dapat hadir bersama dalam rangka melaksanakan Top Scientist Webinar World Class University to Update Technology in Dentistry. Tiada harapan dan dambaan kami melainkan mengharap barakah, inayah dan ridha-Mu selalu menyertai kami dalam kegiatan ini. Allahumma ya Allah, ya Rahman ya Rahim, limpahkanlah kepada kami kekuatan, kesehatan serta keselamatan agar tidak ada hambatan sesuatu apapun sampai selesainya acara ini. Ya Allah, ya Rabbana lakal hamd, jadikanlah pertemuan ini sebagai momentum untuk mempererat tali silaturahim kami dan sebagai media pengembangan kompetensi dan profesionalisme kami dalam rangka meningkatkan kesehatan dan kesejahteraan rakyat dan bangsa Indonesia di masa pandemi dan pasca COVID-19 ini. Jadikanlah acara ini sebagai majlis ilmu yang bermanfaat, yang membuahkan hasil bagi seluruh anggotanya juga bagi masyarakat dan negara. Ya Allah, ya Aziz, ya Ghaffar, bimbinglah kami ya Allah untuk membuka perbendaharaan ilmu-Mu yang maha luas, ilmu yang akan meneguhkan kesucian dan kejernihan jiwa kami, ilmu yang akan melahirkan ketulusan dan kesalehan pada amal kami, ilmu yang akan membawa keberkahan pada profesi kami, ilmu yang akan membawa kemaslahatan bangsa kami, ilmu yang akan mengangkat derajat kami pada kedudukan yang dan martabat yang mulia di muka bumi ini. Ya Allah, ya Rahman, ya Rahim. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. Wa sallallahu ala khairi khalqihi Muhammadin wa ala alihi wa sahbihi ajma'in. Walhamdulillahi rabbil alamin. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you to Ustadz Fajar for the prayer. We hope that today's webinar will be carried out smoothly. Ladies and gentlemen, without further ado, we welcome to the first session's presenter, a presentation from Professor Dr. Tanapum Osatanon, DDS PhD from Chulalongkorn University, Bangkok, Thailand, that will be moderated by Professor Dr. Gigi Ameta Primasari, Dr. Gigi MDSC MKes SP PMM. Professor Ameta is a headmaster of Oral Biology Department, Faculty of Dentistry, University of Sumatra Utara. I will be handing over to the first session to the moderator, Professor Ameta, when you're ready.
Okay, thank you, Intan. Good morning, ladies and gentlemen. Our Honorable Dean, Dr. S.C. Oktiara, Dr. GSP KGA, our University World Class University Chief, Professor Imsar Ambarita, and I would like to welcome you all to this Top Scientist webinar. And in this occasion, we would like to have our honorable guest speaker, Professor Dr. Tanapum Osatanon, DDS PhD from Chulalongkorn University, Bangkok, Thailand. Before the presentation begins, let me introduce our honorable speaker. Professor Tanapum Asatanon received his Doctor of Dental Surgery with honors from Chulalongkorn University, Bangkok, Thailand in 2003. He continued his education of University of Washington, Seattle, USA, and got his Doctor of Philosophy in 2009. Since 2016, he has become a qualified expert in the Center of Dental Competency Assessment and Accreditation, Thai Dental Council. A year later, he has been working for the Committee for Biomedical and Dental Science Examination, the Center for Dental Competence Assessment and Accreditation, Thai Dental Council as a chairperson. He served as a director of Oral Biology Research Unit in Faculty of Dentistry. He served as director of Oral Biology Research, and then the same year, he has elected as a chief of Dental Stem Cell Biology Research Unit in Faculty of Dentistry, Chulalongkorn University, as well as associate editor of Section Regenerative Dentistry, Frontier in Dental Medicine until now. Currently, he has served as Associate Dean in Research Affairs in Faculty of Dentistry, Chulalongkorn University. Professor Asatanon has been doing numerous studies with research expertise in dental stem cell, oral tissue engineering, cell signaling, and bone biology. From this proficiency of research field, he has published extensively in international peer-reviewed journals with more than 1,500 citation, and its index is 20. In addition, he has and his colleagues have written and published several book chapters in international publisher. Now let us hear from Ustad Anom begins his presentation. The time is yours.
Thank you very much. Hopefully everyone can hear me properly. Right. And also thanks Professor Amita for her kind introductions, and also thanks Organizing Committee to invite me here on your webinar of World Class Universities. It's my honor to be here today. And my topic I will present today is about the cell signaling in dental stem cells, which maybe more basic science and not related to clinic that much, but hopefully you can enjoy my talk. I tend to speak quickly, so in the meantime, when I have presentation, if I go very fast, please do not hesitate to let me know. You can raise your hands or turn on the microphone and then let me know. So let me share my slide. So everyone can see my slide, right? Properly?
Yes.
Okay. All right. So the topic I will present today is about cell signaling in dental stem cells. And first of all, I would like to introduce you my school. I'm from Chulalongkorn University, Bangkok, Thailand. So this is the main campus, and the Faculty of Dentistry is over there on the left, which is not in the pictures. We're far away from the main campus, but anyway, we have a close to a shopping center. So if you like to come to visit us, I will be.
Ustad Anom.
Dr. Armita, the signal is not connected. We are still waiting for Professor Tanapum. Yes, here he is now.
I'm now back. So sorry. I'm so sorry.
Okay, Professor.
Okay.
So we're back to the
Welcome back,
presentation.
Charles.
Sorry.
Take your time.
So here again, back to my presentations. Here's our school, and this building is the main clinical building. We have another clinical building over here and over here too. So we have 3 main clinical building, and the small building here is the preclinical building, which location of my lab, and this is lecture hall. And I will be happy to show you around. I mean, if you like come here to visit us at Chulalongkorn University, let me know. I will show you around. So then we go to my talk about the role of cell signaling in dental stem cells. As you may know that in this recent year, the regenerative medicine has been have a lot of influence in terms of clinical treatment. And in the past, the first generation of regenerative dentistry is composed of only biomaterials. For example, using the membrane, using the calcium phosphate materials, mostly are just the use of material base alone. Later, we incorporate some kind of growth factor to promote the functions or to promote the ability of those materials to promote tissue regenerations. And those growth factors that commercially available in the market, for example, endogain, BMP2, PDGF-BB, and FGF2. And these are currently used right now in clinic. The third generation of the regenerative dentistry is incorporation of stem cells. Now the cells is involved, but as you can see here that the use of stem cells in clinical study is very limited. It's still only in the clinical research, not commercially available yet due to the several factors. For example, it's very difficult to culture, it takes time, it's cost very expensive. But again, scientists try to move forward using the cells, not just only using the mesenchymal stem cells, but also construct those cells into the tissue-like structure. For example, use cell sheet or use the 3D construct of those cells as to be make it easier to use in clinic. And the last generation, which now under investigation in the preclinical study, is using the complex of structures construct in the lab. For example, we make a teeth and the bone at the same time and then transplant those units into the patient, something like that. But still, it's far from used in clinic, but it have the idea to use. And hopefully, in near future, we can have that one available in the market, hopefully. So in dental area, we have a lot of cells that can be used as the source of stem cells for regenerative dentistry. We can have the dental pulp stem cells. We can isolate the stem cell from exfoliate deciduous tooth. We can isolate the stem cell from parental ligaments and gingival and also the bone as well. In my lab, we study a lot of stem cells in various types of cells that can isolate from the oral tissues and also the related area, for example, adipose tissues and also bone marrow as well. But all of those stem cells we will talk today is only 2 types of cells. First is stem cell isolated from human exfoliate deciduous tooth, and the second one is about the iPS cell generated from gingival of the isolate from oral cavity. So first of all, I will introduce you about the stem cell isolated from human exfoliated deciduous teeth, which I will from now I will call it as the CHETs. So CHET have been like the cell of interest in the regenerative dentistry because it's very high. It have high proliferative abilities compared to other cell type. It also contains multi-potential differentiation ability, and as you can see, that is isolated from shedding teeth. So the harvesting technique is not that invasive. So you can just extract those shedding teeth out and isolate the cells, and have less ethical consideration compared to the dental stem cells or parental ligament stem cells, because you don't have to really pull the tooth out to isolate pulp tissues and parental ligament cells. So this is self-choice. I mean, in terms of easy access and also have a good ability to use for regenerative dentistry. Here is the characterizations of the cells isolated from remaining dental pulp. of the primary teeth that we perform in our lab. These cells can express various kind of mesenchymal stem cells marker, for example, CD90, CD105, CD44, CD73, and STRO1 as well. But it's lack of hematopoietic stem cell marker, CD45. And once we culture those cells in the specific conditions, for example, in autogenic induction medium or adipogenic medium, this cell can drive to particular lineage that we inducted in. For example, it can differentiate into osteoblasts or odontoblast-like cells as confirmed by the increase of mineralizations in vitro. It can differentiate into adipocytes, and you can see the lipid accumulation inside the cells as shown in red color here. It can differentiate into neuron, into the chondrocytes. And once we implant these CHET cells together with the polycarbolactone hydroxyapatite scaffold and implant into the cover defect of the rat, it can induce bone formation. So all of these characteristics confirming that our isolated cells have the mesenchymal stem cell ability and mesenchymal stem cell characteristic. So in our lab, we study a lot of cell signaling, which is a very boring topic for dentists, as you may know that it's very far from use in clinic, but still, if we understand the basic of this signaling, we can control cell properly, and hopefully it can be used in the future for clinical treatments. This is, there are 5 signaling that we currently use or study heavily in our lab, which is Notch, Wnt, FGF, JAK-STAT pathway, and also mechanosensing pathway. But today I will focus only one pathway, and I select this one to present today because FGF2 commercially available in the markets, and it's been used in clinic for our augmentations right now. So it might be interest of you guys who are in clinical work as clinical in the clinic. So BFGF or FGF2 has been used routinely in culture of human embryonic stem cells. It has to be there in the culture medium because this growth factor, FGF2 or BFGF, can maintain stemness, prevent the human embryonic stem cells to differentiate into specific lineage. So it maintains stem cell properties. So that's why I'm interested in BFGF. And what about the role of BFGF in chats that we're gonna talk today? I will divide into 2 parts. First part is about the role of BFGF to control stemness, meaning that to control the cell to stay in the stem cell stage, not differentiate into particular cell type. And the second one, the second part, I will talk about the differentiation control of BFGF in the chat cells. So first of all, I perform the global gene expression analysis by treated chat cells with BFGF and isolate the RNA and subject those RNA to perform the high throughput RNA sequencing. In this method, we can get 1,000 or 10,000 of genes that differentially express between the treatment and the control. So we can identify the global expression of the genes. Then we can further identify which pathway has been dysregulated using the BFGF. So the result here show that when we divided those differentially upregulate or downregulate gene and we enrich those pathway to find the pathway of interest, we found that the upregulate gene after we treat BFGF in the chat cells are more related to cell cycle control. As you can see, it related to cell cycle, mitotic, M phase, something like that. But for the downregulate genes, most of the genes that downregulate in BFGF treated chat cells involved in extracellular matrix organizations or formations. To confirm that, whether BFGF can control proliferation of chat cells, we treat the chat cells with BFGF and we perform the colony forming unit assay. As you can see here, the blue dot meaning that they have the colony of the cells, and this is the magnification of those colony. Once we treat the cells with BFGF, the BFGF is increased. the colony forming unit ability as confirmed by the numbers is increased, and also when we look into that colony, the cell is more dense, meaning that it's induced proliferations. And this ability can be inhibited by using FGF receptor inhibitors, confirming that the increase of cell number and colony is actually from the influence of BFGF that we add into the culture mediums. Correspondingly, when we do the cell cycle assay, we found the upregulations, we found the increased number of cells in G2S phase and also S phase and G2M phase. And we found the reduction of G0, G1 phase, meaning that cells drive themselves to the cell cycle and have more proliferation. So this results corresponding to what we observed in the colony forming unit assay. One thing I would like to point out here, we inhibit many pathways using a Westcam inhibitor, but we found only one inhibitor that can inhibit the ability of FGF to promote cell proliferations, which is PITK. PITK is the downstream of MAP kinase pathway, which is related to cell proliferation in many cell types. That is about the exogenous treated BFGF into CHET cells, meaning that we add BFGF into the culture medium and see how the CHET cell responds. But in this figure, I show the role of endogenous, meaning that we inhibit the endogenous BFGF in the cells, meaning that we not add anything out, but we inhibit the ability of cell to secrete this or to produce this BFGF using two methods. First one is using HHRNA, which is inhibition at gene level, it's a genetic manipulation, and the other we use inhibitors to inhibit FGF receptor. These two approaches result in the same thing, meaning that whether we genetically manipulations or we use chemical inhibitors of BFGF pathway, we can see the reductions of colony forming unit abilities of the CHET cells compared to control, meaning that endogenous expressions of FGF have the role to maintain or to promote the cell proliferations in CHET cells. So both exogenous and endogenous of BFGF have a crucial role in the modulation of cell proliferation in CHET cells. Not only the proliferations, meaning that when we study stem cells to maintain it in stemness, we look into two parts. First one is the ability of cell to proliferate, and the second one is how cell can maintain in stem cell stage, meaning that it's called stemness maintenance. And we're looking at one of the pluripotent markers of the stem cells, which is called REX1. It's a transcriptional factors, and it's observed this expression of these genes or protein in the cell that stay in the stemness state. The blue color you see here is the staining of DAPI, which is stain for the nucleus, and the green color is the staining for the REX1, which is a protein of interest. When we treated CHET cell with the BFGF, we can find the upregulation, meaning that they have more green color in the nucleus compared to the control, meaning that FGF can promote the expression of REX1. And this ability can be inhibited by using FGFR inhibitors, meaning that we inhibit the FGF pathway, and then the upregulation of REX1 is abolished. So confirming that this phenomenon is occurred to the FGF signaling pathway. And we also show that ATK inhibitor can inhibit the ability of BFGF to induce REX1 as well. And lastly, we use CHX, which is cycloheximidine. It's the chemical inhibitor that inhibit protein translations. When we use CHX, it can attenuate the effect of BFGF to induce REX1 expression in the CHETs, meaning that the ability of BFGF to promote REX1 expressions require intermediate molecules, which try to find which one is it. The one that we identify using the RNA sequencing analysis, we found that interleukin-1 may be the gene of maybe the molecule of interest as the intermediate molecule. To prove that, we culture the cells and treat with the BFGF, and another condition we adding the antibody to neutralize IL-6 activities. So in the condition that we neutralize IL-6 activities, we can find that the REX1 expression, the green color, is less compared to the control which treat with BFGF alone. So confirming our hypothesis that BFGF act to induce REC1 by have the intermediate molecule, and that intermediate molecule might be the IL-6. So for all those experiments, I draw some diagram here to be easier to understand, which is meaning that when we add BFGF into the culture medium, that BFGF bind to the receptor and activate to intracellular pathway. First one is MAP kinase pathway, and this pathway can induce the cell proliferations. Another pathway is ATK pathway, and this pathway induce IL-6 expression, which act as intermediate molecule. Interleukin-6 express, then result in the translations and secrete IL-6 into the culture medium and bind to the receptors and cause the upregulation REX1, which is the pluripotent markers. So this complex regulation here showing how BFGF can maintain stem cell isolated from human exfoliated residue tissues in the stemness stage. The second part, I will talk about the how BFGF can control differentiations. This is very weird idea, meaning that normally one growth factor or one molecule might do one action to the cells, but sometimes it can do a lot depending on microenvironment that we adding those growth factor in the cells. So in terms of differentiation, we focus on the orthogenic differentiation ability of shed cells because we tend to use shed cells as the cell source for bone and dentin regenerations. When we maintain shed cells in orthogenic induction medium to induce them into osteoblasts in vitro, and we observe that these cells have the decrease of BFGF's expression in time-dependent manner, meaning that when it go into commit into the osteoblast lineage, it have decrease of BFGF expressions. To investigate the role of endogenous BFGF on orthogenic differentiation of these cells, we again use two approaches. First one is use the genetic manipulations by knock out the BFGF expression inside the cells, and the other we use the inhibitors to inhibit the FGF signaling pathway. Both of the approaches showing that when we inhibit either inhibit pathway or inhibit the transcription or translations of BFGF, it can increase the mineralizations in sheds. So it's suggesting that the BFGF might have the negative effect on orthogenic differentiation in these cells, meaning that if we downregulate the BFGF, it promotes more differentiation as confirmed by the increase of mineralization here in this picture. Not only the endogenous effect of BFGF, when we add exogenous into the culture medium, OM mean orthogenic induction medium, and OM plus BFGF has reduced mineralization compared to the OM alone. Again, confirm that BFGF can inhibit the expression or inhibit the ability of this cell to differentiate toward the orthogenic lineage. One pathway though that we try to understand is that how BFGF can regulate the mineralizations in these cells. When we talk about mineralizations, it's have to be think about the pyrophosphate and phosphate regulation metabolisms, meaning that when the cells induced into the osteoblast and osteoblast try to precipitate the inorganic compound to form hydroxyapatite in the bone or in the dentin as well, it's required the regulation of this pathway. Normally, it's involved in several of the protein functions. First one is NPP1, which is the enzyme that cleaves pyrophosphate into the pyrophosphate, which is called PPI here. Pyrophosphate or PPI is very potent inhibitor of mineralization, meaning that if we have a lot of PPI in the extracellular space, it can inhibit the precipitations of calcium phosphate, hence attenuate the effect of mineral depositions. PPI have both inside and outside the cells. One molecule we call ANKH, which is a transporter, can transport PPI from inside the cell to outside the cells. And then if the function of ANKH is increased, then we have more PPI outside the cells. When we have more PPI outside the cells, then we have less mineralizations. The other molecule I will talk about is TNAP, or you know as the alkali phosphatase, which is the early marker of osteogenic differentiation. This enzyme can break down PPI into PI. PI is a phosphate ion. So phosphate ion can bind to the calciums and precipitate to be a calcium phosphate and further transform into hydroxyapatite. So the working in concert of this protein can control the mineralization of calcium phosphate outside the cells. And what we study is we try to understand how BFGF can regulate or modulate the expression of these genes and protein in CHET cells. So we treat CHET cell with BFGF and try to find a pattern of ALP expression, NKH expressions, and also NPP and PIT1 as well. BFGF do not have that much role in ENPP expression, but it have the tremendous role in the increase or upregulation of ANKH. And it downregulates ALP expression, meaning that if we have more ANKH, we have this molecule more, then we have more transporter to transport PPI inside the cell to outside the cell, so causing the precipitate accumulation of PPI outside the cells. As I mentioned before, if we have more PPI, then we have less mineralization because this molecule is the inhibitor of mineralizations. And again, at the same time, we have accumulation of PPI, but we have less alkali phosphatase here. So we have less enzyme that can break down pyrophosphate into the phosphate. So we have less PPI to, we have less PI to incorporate with calcium and causing the mineralization. So from this idea, we observe that at later time point when we add the BFGF into the culture medium, we have less PI per PPI ratio, meaning that we have more of the PPI in extracellular sites. So confirming our hypothesis that BFGF may be regulate these three molecules to modulate the mineralization in CHET cells. And as you can see here, when we adding alkali phosphatase into the culture medium, it can rescue the effect of BFGF on the mineralizations. So confirming our hypothesis is maybe the right way to go. So from the beginning until now, I show you that BFGF have a role in controlling properties or controlling behavior of stem cell isolated from human exfoliated deciduous tooth. When we add exogenous or we manipulate endogenous expression of BFGF, it will alter the osteogenic differentiation ability, meaning that if we have more BFGF, it will induce or it will reduce osteogenic differentiation ability. But in the same time, if we add BFGF, it can maintain stemness. It can make the stem cells stay in a stemness state and induce proliferation. So in the future, if we want to use stem cell isolated from human exfoliated deciduous tooth in the clinical treatment, and we have less amount of cells, so we can use the BFGF as the growth factor to add into the culture medium to make more stem cells, and then we can use in the futures. Something like that. That's the kind of idea that we use. We'll use for clinical application in futures. So that has been said. First section is already part. Maybe if I speak too quickly, let me know. I tend to speak quickly personally. So next part, which is the one of the cell that we also investigate in our lab as well is the iPS cells. This iPS cell is derived from mouse gingival fibroblasts. I will talk about the signaling regulations that control the osmotic differentiation of murine gingival fibroblasts derived induced pluripotent stem cells. Induced pluripotent stem cell can be generated from various kinds of tissues. If you look into literature, we can induce this type of cells from fibroblasts from anywhere in the body, even in the keratinocytes, even in the very specific cell type. For example, hepatocytes can be induced to become the iPS cell as well. But the gingival fibroblasts may be a good cell source in terms of dentistry because it's very easy to obtain the sample. You can do the biopsy easily. If you use the pulp tissue or if you use the parental ligament cells, we have to extract tooth and we have to get more invasive technique to pull the tooth out. So gingival fibroblasts may be the easier cell source for use in iPS generations. And as you can see here in this figure, we compare gingival fibroblasts, not we, our collaborations compare the ability of gingival fibroblasts and the tail fibroblasts from mouse. And the result showing that gingival fibroblasts from palatal area have more ability or more efficiency to reprogram into iPS cells compared to tail fibroblasts. And these iPS cells have much higher expressions of marker G related to pluripotent state and also have more expression of the third mRNA, meaning that it can have more property or sameness properties of the iPS compared to the mouse tail fibroblast. And once the iPS cell generated from gingival fibroblast implant into the mice, it can form teratoma, showing that they have more pluripotency to differentiate into various cell type, and it can make a chimera mice when we inject into the embryos of the mice as well. And this work has been done by Professor Hiroshi Ekusa at Tokyo University, our collaborators. So what we done in our lab, we obtain those cells, the mouse iPS cells, and try to induce it into osteoblasts in vitro in the lab. What we do in the lab, we perform what we call the retinoic acid induced method, meaning that we isolate the iPS cells from the feeder cells. We culture it as a floating cells. We call it embryonic body. And we add retinoic acid into that culture to induce the mesodermal commitment. So making these pluripotent stem cells into the mesodermal lineage. And once it commit into mesodermal lineage, we seed those cells again on the tissue culture to make it into osteoblasts. So this is overall technique that we use to induce osteoblasts from the iPS cell that we obtained from Tokyo University. The figure here just to show that using that method, we can induce osteogenic differentiation from iPS cells. As you can see that we have the upregulation of the marker gene related to osteoblasts, RAM2, OSX, collagen 1, OCN, DMP, BMP2, and also the phenotype change. As you can see that we have more and more mineral depositions in the time-dependent manner, confirming that the method we use to induce osteoblasts in vitro from iPS cell is very efficient. In our lab, we focus on 3 signaling that induce osteogenic differentiation in iPS cells. First is hypoxia, second is notch, and the last one is the mechanical force. For the hypoxia and the notch, as you can see here, we published in Journal of Tissue Engineering and Regenerative Medicine and Social Science several years back. When we culture iPS cells in hypoxic conditions, it can induce more osteogenic differentiations as compared to control. You can see here that we have more red color. Red color means we have more mineralizations in the cells, culture conditions. Also, if we use the chemical compound that mimic hypoxic condition, for example, cobalt chloride and DFX, it also can upregulate the ability of these cells to differentiate into osteoblasts as well. And this figure showing that when we seed the iPS cells on jacket1 culture conditions, which is the large ligand, it can induce more mineralizations compared to the control. So implying that both hypoxia and notching can modulate or promote the osteogenic differentiation in iPS cells. So this is summarized of what we found for hypoxia and notch signaling pathway. But the one I will talk today more extensively is the role of mechanical force in iPS cells. Mechanical force has been shown to modulate stem cell properties and also control the differentiation in various cell types. In our lab, we study a lot in PDL cells, in hybrid cells, but the knowledge regarding this mechanical force in terms of iPS cells modulation is not yet identified, or it has been identified, but not so much knowledge out there. This is a figure of the machine that we use to generate the force. The lower part here is the cell culture plate, and the pretzel here is the one that can compress down and give the force to the cells. Using this machine, it can generate a force to the cell without destroy the cell membrane. It can keep the cell intact during the culture conditions. So this apparatus and the use to generate compressive force to the cells has been published before in Journal of Dental Research in 2015. So when we use that apparatus and give the force to the iPS cells for 24 hours, and after that we culture it in osteogenic induction medium, as you can see here that it increased the mineralizations in the force-treated conditions compared to control. Not only the inductions of mineralization, it also induced expression of RUNC2, OSX, and also calcium as well, which is the osteogenic markers, confirming that if we treat the force to the iPS cells, it can induce more osteogenic differentiations. So we try to understand how or which pathway that involved in this mechanism. So we perform a high throughput RNA sequencing. We isolate the cells, we isolate RNA from the cell treated with force, and subject to RNA sequencing, we got like thousands of genes that relate up differentially dysregulate between the force and the control. And among these differentially upregulated genes, we found two genes that interest, which is called MT1, MT2. MT1, MT2 has been shown before to have the role in osteogenic differentiations. And once we treat the force to the iPS cells, we found upregulation of MT1 and MT2. And during the normal osteogenic differentiation of iPS cells, we observed that iPS cells increase the expression of MT2 in a time-dependent manner, so suggesting that the role of MT2 in the regulation of force-induced osteogenic differentiation in iPS cells. But still, we're working on this to try to understand and identify the pathway involved or the downstream target of MT1, MT2 in order to promote or control differentiation of iPS cells into the osteoblasts. But one thing that we interest is that when we analyze those genes that differentially express between the force-treated condition and the control conditions of iPS, we found that the force can upregulate various genes related to cell cycle. For example, CCND1, CCNG1, and also CBK6. So meaning that maybe the force can promote proliferation of iPS cells. So what we do, we just confirm by using the cell cycle analysis. And as you can see here that the force treated conditions have the reductions of G0, G1 subpopulation, but have much more increase of S1, S population and G2M populations. And overall proliferation index is increased in force treated conditions. So meaning that if we add a force into the culture condition of IPS, it can promote proliferation. So this may be good in terms that we have less cells and we want to have more number of cells for future therapeutic approach. We can use a force to induce proliferation of the cells. In the meantime, not only proliferation is increased, but also when we treat the force with the cells, it can attenuate the apoptosis. It have a lot of gene that differentially expressed in P33 pathway, which is a pathway involved in apoptosis. And as you can see here by the staining of annexin V and potassium iodine, the one that treat with the force have lower amount of early apoptotic cells compared to the control. So again, it's kind of like combined together between the effect of the force. So the force induce proliferation of the iPS cells, and at the same time it's reduce apoptosis. So overall, it maybe promote more number of the cells when we culture under the force conditions. This picture try to identify which pathway may be involved in the role of force to induce cell proliferation. So the one signaling pathway that we think of is transforming growth factors. We select this pathway because in previous study of our lab, we study a lot of TGF-beta signaling pathway in human parental ligament cells. So when we apply the force to PDL cells, it upregulates TGF-beta a lot. So in the same time, we observe here, when we add the force to the iPS cells, it can upregulate TGF-beta in both mRNA and protein level, as shown by the PCR here and ELISA and the immunofluorescence staining. Once we inhibit this TGF pathway using the chemical inhibitors, it can attenuate the effect of force-induced CCND1 and CDK6 expressions. So it's likely to confirming that the ability of force to induce proliferation might be occur via the TGF-beta pathway. So hopefully I will not talk too fast, and I can give you the idea what we have done here in our lab in Chulalongkorn University. So today I talk about the study we perform in CHET cells and IPS. Some of those I haven't mentioned here, which we also study in the lab as well. For example, the power stem cells, without stem cells, and SCAP as well. So I would like to thank my funding agency and also the member in my lab here, and our collaborator from Tohoku University, who the one provide us the iPS cells, and our collaborator in University of Paris. And I leave it here just in case that in the future we have collaboration between our schools, so I can put some of your picture over here that we have collaboration with the University of Sumatera Utara as well. And lastly, I would like to talk a little bit. As you may know that we have a very warm welcome from Dr. Atika, who is alumni from our MSD program in allergy at Chulalongkorn University. She is very nice person and very diligent, and she contributes a lot to our program here at Chulalongkorn University. She graduated MSc, master program, with one publication which is very high renowned journal of Parenterology, which now impact factor is 6.9, which is very high impact factor. I'm very grateful that we have her here in our school for the program as well. She worked under the supervision of Associate Professor Piyama Samdetchakanjanakit. So if one of you might be interested in this kind of work, please feel free to apply to our program in MS, PhD, or postdoctoral training in allergology. We also offer Chula Tohoku double degree PhD program as well. And I would be very happy to consider the applications for scholarship if you get recommendation from Dr. Atika. So if you have any question, please directly talk to her, and I'll be happy to consider all the applicants from University of Sumatera Utara. Thank you very much.
Okay, thank you. Thank you for your information and interesting presentation, Professor Osatanon. Now, all right, ladies and gentlemen, we will come to Q&A session. There will be 2 sessions maybe, and each session is for 2 persons. Please don't forget to mention your name. Any question? Yeah,
Am I talking
obviously,
fast?
yeah. Yeah, obviously we are behind your study, Prof. Asatanon, because cell signaling quite heavy for us, But maybe we'll have a collaborative research with you in Chula and USU. Maybe Atika will have a question for Prof. Asatanon. Or you can put in a chat any question. In bahasa juga boleh. Bahasa Indonesia, of course.
Oh, sorry. Okay,
Yeah.
I just tried to unmute my microphone, but at the earlier I cannot. But finally I can speak actually. Sawadika Achan. Thank you. Okay, thank you for your participation in our webinar. So actually, I'm not that excellent. You, I'm just an ordinary student. Okay, but once again, thank you very much for allowing us to have you today. Maybe can I have a simple question for you, Professor?
Sure.
It's a simple question. So, you talk about you talk about cell signaling, and we have known that there are a lot of cell signalings, especially in dental stem cell. So, I just wonder, how did you determine? I mean, when we want to know the pathway in a dental stem cell, I mean, when you want to see the osteogenic differentiation of a certain cell types, let's say maybe sheds, and how did you how did you determine the possible pathway that is involved in that differentiation? I mean, how did you select the possible pathway? Because there are a lot of, so it takes time to try one by one signaling. So yeah, maybe you can
understand the question.
Yeah, I understand. It's very good questions indeed, because like you say, there are tons of signaling pathway involved in the cells control. I show you that I interested in 5 pathway, but actually they have like so many pathway that involve in the regulation of stem cell behaviors. Your question is very interesting, and it's a very good question. In the past, we have very difficult time to identify the candidate pathway, because we don't have the tools, the good tools to identify which is the candidate pathway. So we have to do one by one. So just read the paper and think that this one might be involved in the control of osteogenic differentiation. So we select one by one. But in these recent years, we have very good tool. We use the RNA sequencing technologies. So we can screening the expression of the genes in one time, like 10 or 10,000, more than 50,000 genes in the same time. So in order to identify the pathway that may be involved, we culture the cells in osteogenic induction medium to try to drive the cells into the osteoblast or the blast or the target cell that we want. And then we compare using this gene expression profiles using RNA sequencing technique. We can compare which gene related pathway that can which have higher expression or lower expressions compared to the control. So we group that together and do the enrichment analysis, and it will identify which pathway that may be involved in this particular phenomenon, and then we go from there. That is our approach right now in our lab. But still, if we have tons of questions, I mean, if we have tons of gene expression in the same times, we have to identify one by one. So it's come back to the routine method that we have to like select one by one signaling pathway to dissect how that pathway particularly regulate stem cell behavior. But at least we have like the candidate pathway that we try to look for, something like that. Hopefully answer the questions.
Okay, thank you. Thank you very much, Achan. And before going, Prof. Amita, I just want to say hello to my advisor. Actually, she is attending and joining us here. Sawadika, Achan, maybe you can hear my voice. So, yeah, welcome to our webinar this morning, Achan. Okay, thank you very much for your attending this morning. That's all, Prof. Amita. Itu aja dari Tika. Terima kasih.
Okay, thank you, Atika, for your question and make this conversation interesting, more interesting. You know, Prof. Asatanan, maybe here many of clinicians join the webinar. So if you have simple words about the cell signaling, how you make many pathways, cell signaling for the objective you're confirming for your hypothesis in your study. So I mean, is that the pathway all you have to check, or the obligation is only several pathway?
It's very difficult question to answer. I mean, because well, in terms of how to use in clinic, we have to consider that the study in the lab and the real life treating patients is totally different.
Yeah, of course, yeah.
For example, BFGF, which I present to you today, that I show that when we add BFGF into the stem cells, it can inhibit the differentiation into osteoblasts. But actually, BFGF has been commercially available in the market as the growth factor to promote bone formation in oral cavities. And it can be sold in the US and used in clinic right now. And the results showing that when we inject the delivery system of BFGF into the alveoli, it can promote bone formation. So totally different from what we found in the lab, meaning that when we study in clinic, there are so many factors involved. Need one cell type, but also the serum of the patient, genetic background of the patients, and tons of things. So, but how to say, signaling pathway in the lab may be the first step. Try to understand how this molecule or this growth factor can participate, control specific response. Then we can further this knowledge to the clinic to make it more useful in terms of treating patient in the future, something like that. So I cannot specify specific pathway right now because it's more far away from use in clinic.
Okay, thank you, Prof. Asanan. So any other question? Any other question about the cell signaling? Maybe Prof. Timurni?
Yes, thank you, Prof. Amita.
Thank you.
Prof. Tanapam, I want to ask you, how does epigenetic modification affect a cell signaling?
Could you say that again?
About epigenetic modification. Because in the regeneration of the pulp, there are coordination between TGF-beta, cellular signaling, and epigenetic regulation to cell differentiation.
Yeah, and I this is quite hard question. I say, oh, you guys ask a very hard question to me, a lot. It's very complex. What I show you today is only the study in the gene expression level, but like you mentioned, the gene expression level can control by epigenetic level as well.
Yes.
we have less knowledge for that. We try to do more in-depth, further step to see the epigenetic control, how those can regulate the stem cell property as well. But it's very difficult to study, because when we perform epigenetic study, it's not only particular one genes. It can manipulate the whole sequence, the whole genome changing. So it's very difficult, but it's very great questions. And if we can like connect it between the gene expression level and epigenetic expression level, the control. So if we connect these two levels together, we can like further or can control tightly about how to drive the cell into particular cell type that we want, or specify the function of cell that we want using these two methods together. That's excellent questions, but it's very hard question to answer. Thank you very much. Anyway.
Okay, thank you, Prof.
Prof. Osatanon, how many years have you studied about the self-signaling that you have talked about?
You mean how many signaling?
No, how many years?
Or years?
Years.
I would say 10
study.
10 up, 10 years up.
10 up, yeah, 10 years up, yeah? Okay,
Yep.
then. Any question? Any other question, please? About the time, I think time already 5 minutes more.
Maybe Prof. Amita, one more question.
Yeah,
before
please.
closing.
Yes.
Okay, Prof. Do you think that one signaling pathway can be involved in? Can be involved in other type of stem cells? One signaling pathway is not only involved in one type of cells, but can be involved as well in other type of cells. Do you think? Do you think so?
Yeah, sure. One signaling pathway can control various cell type, but again, specific cell type may be respond differently to the one signaling pathway. For example, if we treat BFGF in chat, it may inhibit autogenic differentiations. It may promote stemness maintenance, but once we treat BFGF to other cell type, it can totally have different effect as well. So it depends on cell type and cell source. In the same time, one pathway can control the same response across the cell type as well. For example, notch-link pathway, when we treat with the chat or dendritic stem cells or parenchymal ligament stem cells, it can induce mineralization altogether, all the same among those cell source. So we have to identify one by one. You cannot say in global, because each signaling pathway and each cell type have particular or specific modulations. Yep.
Okay, thank you very much, Professor.
Okay, ladies and gentlemen, finally we come to the end part of our webinar. We would like to thanks again to Prof. Osatanon for interesting talk, and also thank you for the audience for active participation. Hopefully this webinar will be beneficial for everybody for in the future also, of course. Thank you. So thank you very much.
Thank you very much. It's an honor to be here. Thank you very much.
Back to the MC. Done.
Thank you very much, Professor Tanapum Osatanon and Professor Ameta, such an inspiring presentation today. And the certificate for this session will be given by Dr. Olivia to Professor Tanapum and Professor Ameta as an appreciation from us.
Thank you, Prof, for your great presentation. I would like to give token of appreciation for our speaker, Prof. Dr. Tanapum Osatanon, DDS, PhD. Thank you, Prof.
Thank you very much. My honor to be here.
And okay, Prof. And I would like to token of appreciation for our moderator, Prof. Dr. Ameta Primasari, Dr. Gigi, MDSC, MCAS, SPPMM. Thank you, Prof.
You're welcome, Olivia.
Okay, back to MC.
Okay, thank you so much, Dr. Olivia and Professor Tanapum Osatanon and Professor Ameta. Ladies and gentlemen, we have reached the end of the first session. Now we will be having a break and we will continue the second session at 1 p.m. All the participants are advised to attend the second session 10 minutes early. Thank you for your attention. See you in the second session.
North Sumatra, a sign of God's love given with full affection, a piece of heaven attached to the earth, home of the world's largest ancient volcanic lake that holds 1,000 legends and mysteries, a land for diverse cultural conformity where civilizations live in harmony, a place of dignified tolerance where the seeds of hope are planted, grown, and reaped for prosperity. The genuine miniature of Indonesia.
By declaring the tagline transformation towards the ultimate, Universitas Sumatera Utara is ready to transform into an international standards university with excellent local characteristics. I am Murianto Amin, presenting you Universitas Sumatera Utara.
Starting from Universitas Sumatera Utara Foundation, which collected moral and material support from the people of North Sumatra and Aceh, on August 20th, 1952, the Faculty of Medicine was successfully established. USU was inaugurated as the 7th state university in Indonesia by the first president of the Republic of Indonesia, Sukarno, on November 20th, 1957. Located in Medan City, North Sumatra, USU consists of 2 campuses located in Jalan Dr. Mansur and Kuala Bekala. USU has 15 faculties, 1 graduate school with 163 study programs, and 1 experimental garden covering an area of 300 hectares located in the Tambunan area, Langkat Regency. USU has the vision of becoming a university that has academic excellence as a barometer of scientific advancement that can contend in the global world. Carrying a new spirit, transformation towards the ultimate supports the entire USU academic community to make significant changes to their mindset and work culture per the demands of bureaucratic and technological acceleration. Transformation towards the ultimate is indicated by a series of transformations in various sectors. The transformation is realized through collaboration and synergy between the academic community and various parties to create excellent higher education services implemented through Tri Dharma. Campus digitization is an absolute must to provide the best service. Various digital services are provided by USU, such as ASA, e-office, e-agenda, PPID, and also telemedicine. Telemedicine provides easy access for medical treatment to the public through their gadgets. USU has North Sumatra, a sign of God's love given with full affection, a piece of heaven attached to the earth, home of the world's largest ancient volcanic lake that holds 1,000 legends and mysteries, a land for diverse cultural conformity where civilizations live in harmony.
Fakultas Kedokteran Gigi USU merupakan fakultas kedokteran gigi pertama yang berada di luar Pulau Jawa, yang diresmikan pada tanggal 3 November 1961. Kuliah pertama kali dimulai pada bulan November 1961, di mana masih meminjam 42 buah kursi pada Fakultas Pertanian. Jumlah angkatan pertama berjumlah 28 orang yang terdiri dari 15 pria dan 13 wanita. Bulan Oktober 1962, Fakultas Kedokteran Gigi mulai menerima dosen tetap, yaitu Dr. Gigi Nyonya Mundia Mohtar dari Universitas GM Yogyakarta, memberikan mata kuliah dental material dan propedus teknik. Kemudian, beberapa saat, pemerintahan Jerman menyumbang alat-alat atau unit gigi dengan memenuhi beberapa syarat dan ketentuan, berupa menyediakan tanah dan membangun gedung-gedungnya. Fakultas Kedokteran Gigi berkembang dengan pesat, terutama dengan bantuan dari Lord Edibi. Pada bulan November 1987 sampai 1989, baik penambahan peralatan dental klinik, penambahan gedung perkuliahan, serta bantuan pendidik staf pengajar. Dekan Fakultas Kedokteran Gigi USU dari masa ke masa, periode 1961 sampai 1962, Profesor Dr. Mustopo. Periode 1962 sampai 1964, Profesor Ocilin. Periode 1964 sampai 1966, Dr. Gigi Nazir Alwi. Periode 1966 sampai 1970, Dr. Gigi Nyonya Mundia Mohtar. Periode 1970 sampai 1974, Dr. Gigi Pitojo Suparto. Periode 1976 sampai 1981, Dr. Gigi Rustam Latif. Periode 1981 sampai 1987, Dr. Gigi Mansur Tanjung. Periode 1987 sampai 1994, Dr. Gigi Nyonya Tuti R. Ciptono, spesialis bedah mulut. Periode 1994 sampai 2001, Dr. Gigi Sori Muda Harahap, spesialis bedah mulut. Periode 2000 sampai 2010, Profesor Ismet Daniel Nasution, Dr. Gigi PhD. Periode 2010, Profesor Hadi Naziruddin. Dilanjutkan Profesor Dr. Gigi Treliabul, Magister Kesehatan Spesialis Radiologi Dental Konsultan. Dan pada saat ini Dr. Gigi Esi Oktiara, Spesialis Kedokteran Gigi Anak. Seiring berjalannya waktu, Fakultas Kedokteran Gigi USU telah memiliki sejumlah gedung, meliputi gedung perkuliahan yang dilengkapi dengan fasilitas media pembelajaran berupa whiteboard OHP, LCD, dan alat pengeras suara. Ruang serbaguna Nazir Alwi. RSGM USU. Klinik integrasi. Ruang CBT dan OSCE. Masjid,
dan ruang multipurpose. Seiring berjalannya waktu, Fakultas Kedokteran Gigi kini sudah memiliki 11 departemen dan 6 prodi. Dan pada saat ini Fakultas Kedokteran Gigi USU sedang berulang tahun ke-60. Selamat ulang tahun Fakultas Kedokteran Gigi USU. Fakultas Kedokteran Gigi tumbuh, bersatu, dan tangguh.
Dari sana nggak ada.
Thank you.